I am Roser Vento, and I am a group leader at the Sanger Institute. My team uses genomics, computational tools, and in vitro models to try and understand how tissues form, and in particular, we are interested on how cells talk to each other to form tissues. Yeah, I think there are two things that are quite unique. One is that it's a relatively small conference, and that's really nice because you get to talk a lot with speakers and with postdocs and the students from other labs and really make a sense of having a community. We are quite an open, collaborative community. It's quite a unique conference in the sense that we are quite of a multidisciplinary field and we came from different scientific backgrounds. The single-cell field started as a very small kind of community, but now, more and more people are using it, and we really need these type of conferences to get to know each other, to talk and understand. And what it is quite unique of this one is that it's quite a relatively small conference with plenty of time to catch up with people, to discuss your research. So it's really a unique opportunity to networking with others. I think we are at the moment where we are generating a lot of data from using different technologies, and really all of this is key to understand what a cell is, but also how it interacts with its environment. And we are at the moment of where it's super exciting to be in this community, and from the talks we can see more and more modalities kind of coming, even those that will allow us to understand the mechanistic forces and how it has an effect on the cell. And the other thing is how the computational analysis and new machine learning tools are essential to really make sense of all this data that is being generated. So overall I feel it's quite of an exciting time to be, and we could have seen this in the presentations. And the generation again of plenty of data with at the beginning, it all started with just analyzing the transcriptome as a proxy to understand what the cell was doing. So in a way, single-cell genomics was equivalent of single-cell transcriptomics. And actually now we are just measuring a lot of things at the single-cell level, and it's allowing us to understand all these layers of regulation when integrating it. So in a way, both generating data and being able to analyze it. Oh, it feels amazing. I think on one hand it's good to get to talk after the presentations with the speakers, and a lot of you get to understand better the work. Then I also feel that people, and maybe I include myself, you feel more comfortable sharing unpublished data, because there is also a debate after this that you receive feedbacks from others. So I think it's quite amazing. And then also people like kind of staying in the talk, and is more focused, because you just, when you are at home, you start with the emails and other things, and here it's just the talk, and you get to focus on this. So it's been amazing. Well, it's amazing. Yeah, I think it's as a supervisor, is the best thing that could happen, seeing how these people shine in when they present, and it's really their work. So I think it's also an exciting opportunity for them to present and see, like, when you are in the lab, you only see the negative things sometimes, and it's a way to say, oh, I have done all of these and I can present it. I think it is it is quite amazing. In our field it's really important, the communication and being able to simplify your message so others can understand you. And these presentations, you need to make an effort for doing that. So I think it's really important for students and postdocs to do this exercise, which is actually quite difficult. I think, as I was saying before, it's super exciting field to be in, because we all, or the majority of people, ask questions about, want to understand what are they doing, and the single cell is allowing us to characterize all of these. I think it's a unique opportunity to understand where the field is going, and again, because this is just quite relatively small, and we are in the campus, which is an amazing environment, you get to interact a lot with people. And I think that's really key. This networking can shape a lot of your future directions. And you can see a lot of PhD students, postdocs talking with other group leaders that are super busy because here they have the time. So it's really is a quite unique experience. Yeah, I think that this is very important, because there are people who cannot travel, and that could be because of budget reasons. And I mean, there are here we have some program to support low-income countries, for example, but still there are people who cannot travel. And then also because, for example, the restrictions of the pandemic. So it really, or maybe because they are sick. And so it's really a way to make the conference accessible to everyone, so more people can join and enjoy the beauty of it. The good thing about being in person is that you get to interact with people, so you can have kind of the best of both worlds. If you have the opportunity to come, then it's the best, but then if not, you can still enjoy it. I have been in other conferences that are hybrid, and this one is really one of the best ones on this sense, on the technological setup as well, because Welcome Connecting Science is really making a huge effort on making all these transitions between virtual and people attending quite smoothly. So you, as you were saying, you can interact with the speakers, even if you are not on site, through the questions, and people will ask those questions. So it's quite a unique setup.